real. For people that have heard this, my apologies. Welcome to AP Bio YouTube review number 3. This is Big Idea 3, information transfer. So there's going to be some genetics and some cell cycle. We're going to talk about cellular communication and a very little bit about organismal communication. I am Stephen Traphagen. I teach AP Biology in the suburbs of Chicago. And let's see, a couple of announcements before we start. If you haven't yet, check out the review schedule. There's a link there in my slides. If you are watching this on the YouTube, you'll see in the description of this video a link for the session materials that has a PDF of all of these slides and another document I'm going to talk about in a minute. Those are also available at that bit.ly link. It's bit.ly APBio17 vid3. Okay, I'll talk about College Board learning objectives in a little bit, and if you want to submit for future videos, there is a link there and lots of other places. The sooner you get those questions to the teachers who are going to do the videos, the better your questions will be answered. Okay, so Big Idea 3 has a lot of different things in it, and I tried to organize this according to questions that I've gotten, but also in an order that makes sort of logical sense. And so this video is going to be in 4 parts. Part 1, classical genetics, so the stuff we knew about or learned about before we had the technology to look at DNA, RNAs, and proteins. Part 2 then is the molecular genetics, the more modern stuff, including a very little bit of gene regulation, but a caveat at the beginning of this video. I am going to ask Ryan to do some of the gene regulation stuff next week in complex topics, because Big Idea 3 was really, really big. If you have questions as I go along or I don't answer your question well, please put it in the chat and the moderators will get those questions to me by the end of the hour. Part 3 is cellular communication, and we are going to focus on nerve conduction, a little bit on hormones, and then we'll talk a little bit about how cell signaling affects the regulation of genes. Part 4, organism level communication, so that's the ecological stuff. We'll get there. Okay, so part 1, cell cycle, mitosis, meiosis, lots and lots of questions on that. Then we'll talk about what Mendel learned and try and link that back to cell cycle, mitosis, meiosis, and then some stuff that Mendel would not have understood. Okay, by popular request, the cell cycle, right? So every cell, and for now let's start with multicellular organisms, so all of your cells, right? But every living cell has to go through these stages where let's see if I can do this. So in G1, right, the cell is just doing what it's doing, okay? If it's a liver cell, it's doing what liver cells do, right? It is doing its job, being itself. Then it's going to spend a lot of its time in G1, and then if this cell needs to divide pretty soon, it's going to go through S phase where it's going to make duplicate copies of all of its DNA, right? So every cell in your body, your diploids, has 2 copies of every chromosome, 2 copies of every human chromosome, right? You've got 23 pairs. You got 1 of each of those from your mom, 1 of each of those from your dad. Well, if we want this cell to then make 2 cells, it has to make copies of all of that stuff, right? So if we replicate our DNA correctly, then we go into G2. So now the cell has all the genes it had before, but now the chromosomes weigh twice as much. Okay, and from G2, then we go into M phase, mitosis, and oftentimes cytokinesis. So, okay, student question that relates to this. Chromosomes versus chromatids versus homologs, what's going on there? Okay, so we talk about homologs. These are the chromosomes that contain the same gene positions, but they come from both of your parents. In other words, you have 23 pairs of chromosomes, right? In each one of those pairs, you got 1 of your chromosome 1s from your mom, 1 from your dad. 1 of your chromosome 2s from your mom, 1 from your dad, okay? So thinking about this in another way, trying to be topical. We'll see how long this lasts, right? But for you basketball fans out there, the Oklahoma City Thunder have a point guard named Russell Westbrook. He's had a very good year. And the Houston Rockets have a point guard named James Harden. He's also had a very good year. Both of these teams have a point guard position that might win the MVP, right? So we can think of these as homologous players. They play the same position for opposing teams. They might be matched during the game. Doesn't make them the same person, right? So that's the idea here. We inherit our chromosomes in pairs. Okay, you have a pair of chromosome 1s, a pair of chromosome 2s, etc. Okay, so that's the homologous pairs. What about chromosomes versus chromatids? Okay, in G1, this thing on the left, we call this a chromosome. After we have S phase, right, if you're going to split things up, you need to make sure each cell gets a complete copy of everything. So after S phase, we have chromosomes that weigh twice as much as they did before. So this thing on the left, we call it one chromosome. This thing on the right, we call it one chromosome. That seems kind of confusing, right? This is twice as much stuff, so we want to call it two. That's still one chromosome. So let's call it a duplicated chromosome, so we can still call it just one thing, right? But if this is a chromosome and this is a chromosome, we need a name for what the half is. We call those sister chromatids. So when we start talking about mitosis and meiosis, and we talk about what we're splitting, right, that's where we're going to talk about homologous pairs of chromosomes versus separating chromatids. Okay, so back to the cell cycle. All right, so I take my diagrams and I put them in the cell cycle. In G1, you have, if you're us humans, you have 23 pairs of this. Okay. Quick interruption, sorry. Let's see what my stream looks like. Sorry, looks like we're not seeing all of the slides. Apologies, give me just a second. Okay, we should be live again. Let's make sure that this works. All right. Make me smaller. Transition. Okay, here we go. So, cell cycle. Right? There are these proteins that control every cell's progress through the cell cycle. We call them cyclins. Okay? They interact with enzymes called cyclin-dependent kinases. The idea is these complexes, these proteins, tell the cell where to be in the cell cycle. So, things you should worry about for the AP exam. Things that tell the cell where to be in the cell cycle or when to move on to the next stage. Cell size. Am I big enough to divide? Are there external cues telling a cell that they should be pushed into mitosis or they should stay where they are? DNA replication. Did your DNA replicate correctly? Right? So if I look at this graph in front of me, cyclin D is this clock that governs the entire cell cycle. In green, that's cyclin E, looks a lot like something that's going to push a cell from G1 into S, right? Cyclin B in orange starts accumulating in S, and when its concentration in the cytoplasm is high enough, that pushes a cell into mitosis. Okay, so now let's think about what do we need to worry about for the AP exam, and this is why I shared with you those that document of the learning objectives. So the learning objectives are what the College Board gives us, and they tell us exactly what you'd be expected to do on the exam. So what are the test questions going to be? Student can make predictions about natural phenomena occurring during the cell cycle. So making predictions, what do you think might happen if DNA replication did not proceed correctly, right? Or what do you think would happen if a cell in G1 has not grown enough to successfully split? Then you need to be able to describe the events that occur in the cell cycle. So if I tell you a cell is in G1, do they have single chromosomes or do they have those duplicated chromosomes? When do the chromosomes double in size? Okay. All right. So it's been a long time in cell cycle. Let's talk about mitosis, where we had a whole bunch of questions from lots of people. First, what mitosis isn't. Mitosis is not cell division, and mitosis isn't DNA replication. Okay, DNA replication happens way back in S phase. So, what mitosis is division of the nucleus. Okay, so we need to think about what needs to happen for the nucleus to divide. And so, starting here, I've tried to use figures as much as possible from the popular AP Bio textbooks. So whether that's Campbell or Hillis, I'm hoping that you'll recognize a lot of these representations. We're going to get to a mitosis-meiosis comparison, but let's just right now start with mitosis. Okay, and so we see you start with a set of unduplicated chromosomes. So this is a G1 cell. Okay, we have 1, 2, 3, 4 chromosomes. So in this case, a diploid cell, 2N is 4 chromosomes. We go through S phase, that gets us to G2. We now have duplicated chromosomes. So here we have 4 chromosomes, and here we have 4 chromosomes. Okay, they just weigh twice as much. All right, from here we have these duplicated chromosomes. They need to be lined up single file, okay, along the metaphase plate. Now again, in AP Bio, this is different than it was in the old days. You don't need to know the names of the phases, so you don't need your PMAT flashcards. What you need to know is what happens in those phases, right? Meiosis is a continuous process. So whether something is prophase or prometaphase or metaphase, to be honest, my background's in genetics. I don't care that much. I just want to count chromosomes and make sure that the daughter cells get what they need. So we have our 4 duplicated chromosomes. They line up single file. This is really important. If they line up single file as the spindle fibers attach, what those spindle fibers are going to separate, they're going to rip those sister chromatids apart, right? So what you end up with is 2 daughter cells that each have 4 chromosomes, just like we started. We have 2 big chromosomes, one from dad, one from mom, and we have 2 small chromosomes, one from dad, one from mom. So these 2 cells are exactly what we started with, okay? Here was our cell in G1 before it did S phase. We've got 4 unduplicated chromosomes. We do a bunch of things, right? Here's our G2. I cut out the middle steps. What's important is you start with 4 unduplicated chromosomes. You end with 4 unduplicated chromosomes, okay? All right, we're going to get to meiosis. But I think sometimes doing mitosis and meiosis back-to-back causes more confusion than it helps. So we're going to go into what Mendel did with his pea plants, because though he lacked the technology to see inside the cells and see the molecules that we know about, Mendel really discovered the pattern of meiosis. Okay, so I think it's important for you to understand his 2 big experiments, the monohybrid and the dihybrid cross, and why those experiments led him to the conclusions that they did. Okay, so the monohybrid cross, he always started with true breeding plants, right? He had this idea that living things had these factors, right? We call them genes or alleles, but they had these things, these packets of inheritance. He always started in his parental generation with things that were pure for whatever factor he was studying. So purple, when he crossed that plant to itself, it always made purple. White crossed to itself always made white. And so when he crossed purple to white, all of their offspring were purple. So this is where he came up with this idea of dominance and recessiveness, right? Dominant alleles are the things that show up in a hybrid, and recessive alleles are the things that hide in a hybrid. So when he talks about the monohybrid cross, what we're really talking about is this cross here. these are hybrids, those F1s. They're purple, but we know they must have some white factor hiding in them because they got something from that parent. Okay, so the monohybrid cross, when you take those hybrids and you cross those together, what he found was in the F2 generation, in this generation here, all of a sudden, white came back. Okay, and so if white was invisible here in the F1s and it became visible down here in the F2s, okay, then there's no such thing as skipping a generation. That white allele was carried through, but the pattern, the numbers tell us something, or at least they told Mendel something. So the monohybrid cross show that each parent donates one of two factors, we call them alleles, to their gametes, to their egg and sperm cell. He called this the law of segregation. Each parent gives one of its two factors, one of its two alleles to the offspring. Okay? And in a way, he discovered the idea of diploid organisms and haploid gametes. We have twice as much genetic material as we give to each of our offspring. So the monohybrid cross, I should go back before we get to the dihybrid. The monohybrid cross led Mendel to this idea that things are inherited discretely, right? You're not a mix of your two parents. Your parents pass these discrete factors to you, and we can only pass one of the two we get from our parents. Okay, so that's his law of segregation. We're going to come back to that when we talk about meiosis. All right, the dihybrid cross, his second big experiment and what he learned. So now he's going to take true breeding parents, just like he did before, but now we have pea plants, and he's going to consider two traits. What color are the peas, and what shape are they? Are they round or wrinkled? And so he took a true breeding round and yellow pea plant and a true breeding green and wrinkled pea plant and crossed them. And the question he's trying to answer here is, if I inherit the trait for round, does it make me more likely to inherit the trait for yellow? So you can see left and right here the two hypotheses. Dependent assortment. Yellow is going to go with round and green is going to go with wrinkled. And then there's this independent assortment. What if they get shuffled every generation? What if inheriting one thing doesn't affect the way you inherit the other? Just like he did in the monohybrid cross, his F1s are hybrids. In other words, they have yellow and green factors, and they have round and wrinkled factors, and they show one trait over the other. So in this case, they show round and smooth. Okay, so round and smooth are dominant. If these genes assorted in a dependent fashion, in other words, round and yellow always went together, and green and wrinkled always went together, he'd get the same 3-to-1 ratio that he got in his monohybrid cross. You either look entirely like one parent or you look entirely like the other. But that's not what he saw. So this is where we get the 9-to-3 to 3 to 3-to-1 ratio, right? It turns out that your probability as an offspring of inheriting round doesn't have anything to do with your probability of inheriting yellow versus green. So the dihybrid cross shows that inheriting one allele does not affect your probability of inheriting something for the other trait. He called this the law of independent assortment. So yellow and green assort totally differently from round and wrinkled. So this is Mendel's idea that genes, he called them factors, but genes get inherited independently. You'll notice there's a asterisk next to that. We know that's not always true, right? And we can talk about linked genes in a little bit. I promise we will. Before we get into that more modern stuff, another student question: what is codominance? Okay, and so this is a good segue into how these factors actually work and what they do. So remember back when we talked about homologous pairs, right? Well, as humans, we each have a pair of chromosome 19s. You got one from your dad and you got one from your mom. So the gene for blood type is located at one particular position on chromosome 19. and this individual got a type B allele from mom and a type A allele from dad. And so blood type is a good example of a codominant mode of inheritance, right? So blood type is you make these little self-identifying proteins on the outside of your red blood cells, and in this schematic, if you inherit the type A allele, you make these little pink circle proteins, right? This is part of your body's way of identifying your own cells, okay? So your immune system doesn't kill your own cells. So if you inherit blood type B, instead of making pink circles, you make these little green diamonds. But in this case, because we've got one copy of the green diamond allele and one copy of the pink circle allele, this person makes both of these proteins. So that suggests that both of these instructions are being used, okay, which is a good segue into the transcription and translation we're going to talk about in a little bit, okay? So before we get into that, we need to go back to meiosis, and we're going to try and talk about how what Mendel did and meiosis are related. Okay, so here's the diagram we started with, and now we're going to cover up mitosis. And so we're going to start with the same things we started with in mitosis. We're going to start with a diploid cell in G1 with unduplicated chromosomes. We're going to do S phase and G2. We're going to end up with our duplicated chromosomes. And so now in meiosis 1, we get this really weird and interesting thing where the chromosomes line up by homologs. So the chromosome 1s line up next to each other, the chromosome 2s line up next to each other. They line up by size so that matching genes from mom and from dad are right next to each other. Now remember, this, when we talk about the chromosomes from mom and dad, these are your parents' chromosomes, and this is what's happening as you are making your gametes, egg or sperm cells that you might pass to your kids. So we're talking about what you got from your parents and where it's going to get passed to your kids. Okay, so the biggest difference between mitosis and meiosis is right here. Because the chromosomes line up double file in these homologs instead of single file, we're going to separate something else in meiosis 1 compared to what we did in mitosis. Now, in prophase, if you notice right here, these two chromatids are hanging over one another, right? So this is where we're going to get crossing over. I'll talk about that in a little bit. So in meiosis 1, the metaphase plate, these things line up single file, and so when they're pulled apart, we're not ripping sister chromatids apart from each other, we're ripping homologous chromosomes apart from each other. So now at the end of meiosis 1, each of these daughter cells now has half the number of chromosomes it started with. We started with 2 tall and 2 small chromosomes, right? There's my 2 tall, there's my 2 small. And at the end of meiosis 1, each of these daughter cells has 1 tall and 1 small. The other thing that's interesting about this, because of crossing over, we can't say these chromosomes are from mom anymore. They are a novel recombination, a mixtape, as my students say, right, of what you got from your parents. This is what we're passing to our kids. Okay? So, all right, we now have half the number of alleles that we started with. We've gone from 2n to n. All you need to do now in meiosis 2 is to separate the chromatids. Okay, this is Mendel's law of segregation. When he talked about only passing one factor or another to your offspring, right? This is where it happens. Mendel was talking about metaphase and anaphase 1. So as those homologs are split, you're either going to give blood type A or blood type B, right? But not both. So, okay. In meiosis 2, this is pretty easy. All we have to do in meiosis 2 is take our pair of duplicated chromosomes, separate the chromatids. Meiosis 2 looks a lot like mitosis. Line the chromosomes up single file in the middle of the cell and split the sister chromatids apart. So the thing I tell my students is in mitosis, we start with 4 chromosomes duplicated, we end up with 4 chromosomes single. In meiosis, we start with 4 chromosomes duplicated. We end up with 2 chromosomes that are singles, right? But everything interesting in meiosis happens in meiosis 1, okay? Mitosis makes genetically identical cells. All you have to do is duplicate your single chromosomes in S phase and then split them in mitosis. Meiosis, you have to duplicate your single chromosomes just like you do in mitosis. Meiosis 1 serves to separate the homologous pairs. Meiosis 2, all it does is separate the sister chromatids, which looks a lot like mitosis. Looks a lot like mitosis. So the thing I tell my students is everything you think of as being interesting in meiosis happens in meiosis 1. Okay, you've got the independent assortment. As those tetrads line up on top of each other, they can flip back and forth. Okay, you've got your crossing over. That's where the genetic shuffling happens. Meiosis 2 is really just mitosis of these weird haploid cells. Hey, there it is. That's the difference. Mitosis, those chromosomes are lined up single file down that metaphase plate, and meiosis, they're lined up double file. That's what makes the difference. Okay. All right, one more thing that Mendel didn't really take into account when he talked about independent assortment, right? He said your probability of getting one factor didn't affect your probability of getting another factor. Well, that's a problem because DNA is inherited in these giant packets, right? You have your tiny, tiny little double-stranded DNA wrapped around these histone proteins, compacted into these chromosomes. If we're talking about humans, we're talking about 30,000 genes on 23 pairs of chromosomes. That's way more than one gene on every chromosome. You're talking about thousands of genes on a lot of these chromosomes. So there's no way that each of these genes can get shuffled into egg and sperm cells independently. They're not all going to follow Mendel's law of independent assortment. So it turns out if you have genes that are located close together on the same chromosome, because they're attached to each other, they're probably going to get inherited together. Okay? So the exception to this is because of what's going on in prophase 1 in meiosis, chromosomes can lay across one another and they can exchange pieces. So we see one here. Here's a crossover event. So here you could have, and we've made it easy with this diagram, dad is donating all of these dominant alleles, big A, big B, big C, and mom is donating all of these recessive alleles, little a, little b, little c. So you would expect, if these things are close together, you're going to inherit dad's alleles together or mom's alleles together, right? This is what you're going to pass to your offspring. But if you have this crossover event, you, as you're making your gametes, egg or sperm cells, you can pass big A, big B from dad and little c from mom, or you can pass little a, little b from mom, big C from dad. Okay. What you need to know about chromosomes is DNA is packaged in these giant pieces of DNA wrapped around proteins, okay? And you need to know that that means a lot of genes get inherited in packages, but then there is this crossover business. Come back next week for complex topics for more on linked genes and crossover frequency. Okay, let's move into molecular genetics, right? Let's talk about DNA, RNA, proteins, because Mendel was knocking on the door of this even though he couldn't see any of it. So we're going to start with DNA. A lot of you have seen this, right? Your basic structure of DNA, it's this double helix. But let's look more closely at what this thing is made out of. The basic unit of DNA is the one nucleotide, a sugar and phosphate that go on the outside, and then the base, the A, T, C, or G, that give you the code, right? Okay, so here's our 4 bases. A and G are adenine and guanine. Those are the big ones. Those are the double-ringed purines. And T and C, thymine and cytosine, are the single-ringed pyrimidines. A always goes with T, G always goes with C. How do we know that, you might have asked? Got a student question. This is not a history course. You don't need to know the details of everything that's ever happened in biology. But this is a topic, we call it Great Experiments in DNA History, where it is helpful to understand how we came to understand what's going on with molecular inheritance. So the thing I want to point out when we go through Avery, McLeod, McCarthy, Hershey and Chase, Chargaff, and then Watson, Crick, Wilkins, and Franklin. All of this progress towards understanding DNA as the molecule of inheritance, follows after Thomas Hunt Morgan, who did his work at the beginning of the 20th century, and he won a Nobel Prize for it. So it wasn't a secret. Thomas Hunt Morgan discovered chromosomes. He saw these things in fruit flies, and then it still took 50 years for this to be commonly understood that DNA is the molecule of inheritance. Okay, not on the list. So before we can talk about Avery, McLeod, McCarthy, we have to talk about Frederick Griffith. And he killed some mice with bacteria. So the deal with Griffith's experiment was he had a rough strain of bacteria and he had a smooth strain of bacteria. And it turns out if you inject a live mouse with this rough bacteria, they're fine. It doesn't kill them, so it's not virulent. If you inject a mouse with the smooth strain, the mouse dies. Then what Griffith did was he took the dangerous smooth bacteria and he boiled them, heat killed them, injected them in the mouse lives. But then if he took the dead formerly dangerous ones, and he mixed them together with live, not dangerous ones, and injected the mixture, mouse dies. So Griffith's conclusion was there must be some kind of a factor, something inside those dangerous, dead, smooth bacteria that transferred itself into the rough bacteria and made them dangerous. He called it a transforming principle. What could transform these safe bacteria into dangerous ones even after the smooth ones were dead? So where Avery, McLeod, McCarthy came in is what was the molecule that was that transforming principle? What actually does the change? So they did a follow-on experiment where they wanted to try and isolate, was it DNA, was it RNA, or was it protein? And what you need to know about this is they repeated Griffith's experiment, but they treated each of those cell mixtures with enzymes that would degrade DNA, degrade RNA, or degrade protein. So you could try and figure out exactly which one of these is the one that's doing the transforming, changing the bacteria and killing the mouse. And so if you treat Griffith's experiment and you do protease or RNase, and you essentially destroy the proteins or the RNA of those dead virulent bacteria, it doesn't change the outcome of Griffith's experiment. But this is the key one right here. If you repeated Griffith's experiment, but you treated the dead bacteria with DNase, you destroyed their DNA, then all of a sudden we didn't get a dead mouse where we had before. So the transforming factor, the thing that made those rough bacteria dangerous, it must be DNA. They pretty conclusively proved it. Somehow this was still not widely accepted, so we need to do another experiment. Here comes Hershey Chase. So they used phage. These are viruses that attack bacteria. And they said, okay, viruses are really simple. They're protein coat and DNA or RNA on the inside. Okay, you can almost think of a virus as an M&M, right? The chocolate is the DNA, and then it's got that thin, colorful candy shell of protein to keep it safe, right? So what they did was they grew up a bunch of virus in the presence of radioactive sulfur, okay? And that radioactive sulfur, proteins have sulfur and DNA doesn't. So up here in the pink, we have virus where their protein coat was radioactive and their DNA wasn't. And then they injected that virus into bacteria, and then they waited a little while and they wanted to see were the bacteria radioactive or not. Another treatment, they grew up phage in the presence of radioactive phosphorus. Phosphorus is in DNA but not protein. And then they injected that into bacteria and they wanted to see which bacteria stayed radioactive. So mix the virus in the bacteria. Mix the virus in the bacteria, and then spin it down to isolate the bacterial cells. And it turns out that this is where we had the bacterial cells that contained the radioactivity, right? So what the virus was injecting into the bacteria was in fact its DNA, not its protein. So now we've got pretty conclusive evidence that DNA and not protein is that molecule of inheritance. Okay, we always say A goes with T, C goes with G. This was Erwin Chargaff. he chemically cut DNA apart and then isolated all of the bases. And what he found was however much A there was in an organism's DNA, there was that same amount of T, and however much C there was the same amount of G. So we call this Chargaff's rule. He knew somehow that A and T had to be associated with each other, and G and C had to be associated with each other. So now we know DNA is the molecule of inheritance, and we know something about this code. But if this molecule can't copy itself and be passed from cell to cell to cell, then we don't understand how inheritance works. So the next big step, the last big step, was trying to figure out what is the structure of DNA, what is the shape. And this is where Watson and Crick get a lot of the credit, but of course it wasn't their evidence. Okay, Rosalind Franklin worked with a gentleman named Maurice Wilkins, and they were taking pictures of really small molecules with X-rays. Right? DNA is smaller than the wavelength of visible light, so you can't take a picture of it with a microscope. You got to use X-rays. Now, they took this picture. While they're doing this, Watson and Crick are trying to solve the structure just by building models based on their understanding of the chemical structure of DNA. Watson and Crick weren't getting anywhere. So Franklin takes this picture, and through some shadiness that I will skip for sake of time, Watson and Crick saw this picture, and if you can read X-ray crystallography, this tells you this is a helix. Okay, this is a spiral. So they took her work, ran back to their lab, built their model, and won the Nobel Prize. She was working with a lot of X-rays, and unfortunately she passed away before that Nobel Prize was awarded, and never really got the credit that she should have for this. Okay, so that's how we discovered what DNA structure is. I don't have my students memorize a lot. This is the one exception. You need to memorize this, because this is going to affect DNA replication. This is going to affect transcription of DNA to RNA. DNA is always read in the 3' to 5' direction. So if you're going to make some more DNA in DNA replication, or you're going to make some RNA in transcription, the new thing is always made in the 5 to 3 direction. All right, what does that mean? All right, let's look at DNA replication. So we have here this top strand of DNA is going in the 3' to 5' direction from right to left, like that. And so following this rule, if I want to make some more DNA in replication, I'm going to read it in this direction, and I'm going to make my new DNA in the 5' to 3' direction. Okay, we call this the leading strand. It's nice and simple. As this replication fork opens up from right to left, okay, that top strand is going to keep polymerizing continuously. We have a problem down here though. So if you remember the lagging strand, this is why we have this problem. We still have to follow this rule. New polymers have to be created in the 5' to 3' direction. So to do that though, we have to go opposite the direction that this replication fork is being opened. And so we have to wait until the fork opens a little bit further, until you can get an enzyme in here, right, DNA polymerase, to add some more DNA. So the lagging strand is always replicated in these small little chunks. The leading strand always replicated continuously. Last thing, and the way, if you're having trouble with this 3' to 5', 5' to 3', I always think of the 3' end as the wet paint end of the new molecule. That's the fresh end. Okay. All right, a little bit more detail. So we have down here, nice and simple, the leading strand of replicating DNA. This thing is just being added. Now we have the replication fork going from left to right. Okay, and as this thing is opening, right, this blue protein, DNA helicase, okay, is opening up those hydrogen bonds between the strands. As it's doing that, you're getting replication from left to right. Okay, that's nice and simple, but the top strand is much more complicated because we have to be doing this in the reverse direction. We've just about got enough room here to start doing it again, right? And then you have all these disconnected parts called Okazaki fragments, And then, of course, those things need to be tied together. It's a lot more steps. Okay, if that's true, though, what does this 5' to 3' business even mean? We number our carbons in organic chemistry. It's just a way of talking about what's attached to what. So your nitrogenous base, your A, T, C, or G, is here. Okay, and then we number them counterclockwise. Sorry, that's clockwise. The first carbon, second, third, fourth, and fifth. The third carbon we call the 3' carbon. The fifth carbon is the one connected to the phosphate. So when we talk about things being 3' to 5', we're talking about the direction that the sugar and phosphate are connected and the way that the backbone is pointed. Okay, the reason this matters is because when you look at these two anti-parallel strands, we've got the left side, all the sugars are pointed up, for lack of a better description, and on the right side, they're all pointed down, the 3' to 5' direction. Enzymes are really specific to shape, remember, and so enzymes can only read in the 3' to 5' direction. So an enzyme, if it's reading the left strand, it has to go up the page. If it's reading the right strand, it has to go down the page. Okay. All right. So let's talk about gene expression. Lots of student questions on this. Okay. First, you've seen this before. We're going to go quick. Genes don't actually do anything unless you use those instructions to make something, right? And so Francis Crick called this the central dogma. Remember, not all genes end up being proteins. Some genes code for RNA. But in general, right, we think of DNA being transcribed, being copied into that temporary RNA molecule, and then that RNA is then used to be translated to make a protein based on the DNA instructions. So in general, you have your DNA template, it gets transcribed into RNA, and then that RNA is used as a code to make proteins. This is really different in prokaryotes versus eukaryotes, right? In bacteria, it's really nice. The DNA is just floating in the cytoplasm. So as soon as that RNA comes off the DNA, as soon as it's being made, a ribosome can come in and start translating the protein, right? But in eukaryotes, like us, you have a more nuanced process. That DNA is locked up inside the nucleus. The nuclear pores aren't big enough for it to come out. So your RNA gets made in the nucleus and then translated into protein in the cytoplasm. So this is where you have an opportunity for the processing that we do as eukaryotes that prokaryotes don't. Again, I'm going to refer you to the Complex Topics podcast because gene regulation is going to look really different in prokaryotes versus eukaryotes. Prokaryotes with DNA in the cytoplasm, you can have operons. Eukaryotes, we have all sorts of RNA processing, right? But gene regulation is going to be a lot more difficult because nothing in the cytoplasm can directly touch the DNA. Okay, we're going to zoom in on translation. So mRNA is that single-stranded piece of RNA that carries the message from the DNA to the ribosome. Okay, mRNA is made out of RNA. Ribosomes are made out of RNA and proteins. And tRNA, transfer RNA, is also made out of RNA. So all three of the things in this picture are made out of the same material. That's really nice because the way we get our translation is that mRNA carries this code in these 3-letter sequences called codons, right? It binds complementarily, just like back with DNA, C with G, G with C, and of course RNA can't make T, so we use U instead, A with U. And so what you have is as this mRNA is zipped through the ribosome, the tRNA comes in, docks, and brings whichever amino acid it happens to carry, and you've got a different tRNA for every amino acid. In fact, you've got more than one tRNA for many amino acids. So here's the codon chart, right? We have the mRNA on the bottom binding to this tRNA thing, and then, here we go, we can read, based on our mRNA GAG, we can read what amino acid this tRNA is going to be bringing. So we go to the first base is G, so we're going to be somewhere in this bottom row. Our second base is A, so that's going to put us in this box here. And our third base is G, which puts us down here, glutamate or glutamic acid, right? So if you have 4 bases, 3-letter codons, that's 64 different combinations. We have 64 codons here, but you only have 20 amino acids. So there are a lot of repeats, which is nice. It makes mutation much more tolerable. You can change the DNA sequence so that our mRNA changes maybe from a CAA to a CAG and nothing's going to happen. It's a silent mutation. You don't see it, right? Then we also have some punctuation. Our start codon, every protein synthesis has to start with AUG, whether the methionine stays on there or not. And then every protein synthesis has to be stopped by this signal of a stop codon, either UAA, UAG, or UGA. Okay, so that's the questions we got on genetics. Moving into cellular communication feels like a really big shift. So I'm going to focus on 2 things. We're going to talk about nerve cells, and then we're going to talk about hormones. There's a very little bit on how cell signaling affects gene regulation, but we're going to focus on nerve cells and hormones. Okay, here's your basic structure of a neuron. From left to right, you've got the dendrites, which receive signals. You've got the cell body, which keeps that cell alive. And then you've got the axon that can send signals long distances down to the axon terminals that are going to send the signals to the next cell, whether that's a nerve cell or a muscle cell. So we're going to go through this step by step. First, we need a signal to be received by receptors on the dendrites. Then the cell body decides to do something about it. We'll get an action potential down the axon, and then if we make it all the way down the axon, neurotransmitters released from the axon terminal. Okay. On the ends of your dendrites, there are these tiny little receptors that can receive these messages from hormones or neurotransmitters. I'm going to skip my caffeine addiction for now. All right, and go to the action potential. This is where we got most of our questions, right? Okay, so an action potential is the movement of charge across a membrane. And that movement of charge, you can think of it as electricity, right? This is an electrochemical message that's sent long distances, okay? So in the center here, we've got the trace. We're going to look at what's going on across that axon membrane and how that contributes to those, to this voltage change and sending a message, okay? So, okay, a neuron at rest, we say has a potential of negative 70 millivolts, okay? And I was always troubled by this. We've got a bunch of positively charged sodium outside the cell, positively charged potassium inside the cell. How can we have a negative voltage? Remember, voltage is a measure of difference. If I have $100 and you have $30, you still have positive dollars, but if we're compared, you're minus 70 compared to me, right? So we start out where there's a lot more positive charge outside the axon than there is inside. If we can get just a little bit of change in that charge, about 15 millivolts, from negative 70 up to about negative 55, then all of a sudden these sodium channels will open up. They change shape with voltage. We call them voltage-gated channels. And all of a sudden, this sodium that was bouncing off now starts to rush in. And as that happens, we get a massive uptick in voltage. The voltage goes from a negative difference, okay? Now we have a positive voltage. We got a whole bunch more positive charge inside the axon. Once we get a whole bunch of positive charge, that causes the potassium channels to open and the potassium to rush out of the cell. That gives us our repolarization. We're going back down to a negative charge. Okay. And then we call this the undershoot, right? This potassium channel is slow to close. Okay. It looks like we're about ready We're about back to the resting state. We're about ready to send another action potential. But there's one more problem that we have, and that is a lot of our sodium and a lot of our potassium are in the wrong place. In order for this to be reset, you need your sodium concentration outside the cell, and you need your potassium concentration back inside the cell, which brings us to the sodium-potassium co-transporter and why you need to eat a healthy breakfast before the AP exam. Okay, you can pump sodium against its concentration gradient back outside the cell, and potassium against its concentration gradient back inside the cell, and you can reconcentrate these things, but it's going to cost you a lot of ATP. So to do that, those nerve cells in your brain, when you're concentrating hard on that AP exam, those nerve cells are going to be burning a lot of glucose, right? You're going to go through a lot of ATP really, really quickly, lots and lots of cellular respiration. So unless you eat a breakfast containing carbs, I recommend a bagel and some melon, right? You will get progressively dumber throughout the test as your brain starts to run low on sugar. So there's your PSA. Okay, we got just a couple minutes left. Our technical difficulties put us a little bit behind. So, talk briefly about neurotransmitters. This was a student question. So how do we get from this electrochemical movement of charge down the axon to the release of neurotransmitters at the end of the axon terminals? Well, you've got those voltage-gated channels for sodium. At the very end of the neuron, I thought it was called the button. Apparently, my wife is a neurobio person. Apparently, it's called the bouton. Sounds very French. At the end of the axon, at this axon terminal, you have voltage-gated calcium channels. And as the action potential reaches those voltage-gated calcium channels, okay, those allow the fusion of these synaptic vesicles. They allow the fusion of these little vesicles containing neurotransmitters with the end of the axon and release of neurotransmitter into the synapse. Okay, hormones. What do we need to know? This one is quick. I went back to the learning objectives, right? You need to be able to talk about how cells can talk to each other over distances. So here's one example. This is from that plant hormone POGL, right? Phototropism. It turns out there's something being made in the tip of the shoot that talks to the rest of the shoot. If you cut the tip off, the plant stops moving towards the sun. If you cover the tip, it stops moving towards the sun. If you cover the rest of the shoot, the plant is unaffected, right? So somehow these cells are talking to each other over distances. Two big categories of hormones: protein hormones versus lipid hormones. Okay? They each have their advantage and disadvantage. Proteins cannot pass through cell membranes, okay? So they need a receptor on the outside of the membrane, and they're going to have to use second messengers to get across the cytoplasm, to get their message across the cytoplasm to affect the transcription of genes, right? Steroid hormones, they can go right through membranes, but they don't dissolve very well in the blood because that's mostly water, and they don't dissolve well in the cytoplasm. So they need a chaperone protein to get to the cell, and they need another chaperone protein to get across the cytoplasm. In both cases, though, your goal is to get a signal from outside the cell into the nucleus to cause the cell to increase or decrease the transcription of some genes, right? That's how cells respond to their environment. So when we talked about transcription and translation, the expression of genes, these signals tell cells when to do that, right? Cells are trying to respond to changes in their environment. Okay, last topic we've got, and this one is really quick because nobody asked any questions about this. I just wanted to throw out 3 good examples of things that are interesting about communication at an organismal level to remind you that you should study it because maybe it's on the test, and also you should study it because it is wondrous and awesome, and we need to pay attention to the world around us. So, poison dart frog. Things that are poisonous are often very brightly colored to advertise to predators that they shouldn't be eaten, right? This went very badly last time, okay? So another form of communication, the peacock and sexual selection, right? When a male is showing off all of these awesome feathers, what it's really saying is, my genes are great. I've been allowed to survive to this point in my life. I've been able to make all this stuff. I must have really, really good genes. So, and I'll skip the last one. Okay. We've got probably a couple of minutes left. I'm just going to check in with moderators. Do we have follow-up questions? Okay. One last thing I want to say to everybody as I'm looking at the chat real quickly now. There is a lot of really interesting and important information in the world that is not covered on the AP Bio test, right? So we're not trying to teach you exclusively to the test, but we appreciate this is review right before. So for the next couple of weeks, it can be helpful to you to look at exactly what am I going to be expected to know, right? So that's where I would suggest you go to those learning objectives to see what are the limits of what I'll be expected to know. And the one that I think is a pretty good example of this, that calcium signaling, really, really cool, okay? But I think it's an illustrative example of a membrane protein allowing cells to talk to each other. Okay, that's not something where the details of that process are going to be examined in the exam. Okay. Yeah, good luck, everybody. I think looking really quickly at these questions. Where are we? Okay. All right. I want to respect everybody's time. It is 1:06 p.m. So I think I'm going to sign off for now. Remember, though, we do have 4 more of these review sessions over the next 2 weeks. So if you have further questions, please send them to that question submission link, and other people will follow on after me. Thanks, everybody, for listening.